In the previous video, we explored the structure of the skin. Now we continue right where we left off, shifting our focus from structure and anatomy to function and physiology. The skin serves a wide array of critical functions that support homeostasis, defense, communication, and metabolism. Through specialized sensory receptors, it detects touch, pressure, pain, and temperature, then transmits these signals to the nervous system. This allows the body to perceive stimuli and respond appropriately for protection and adaptation. To make sense of its many roles, we'll divide the skin's functions into clear categories. Among the skin's most obvious roles is protection, which operates on multiple levels. Through integrated physical, chemical, and biological properties, the skin effectively serves as a barrier against external threats. The stratified, keratinized epithelium of the epidermis forms a tough physical barrier against mechanical injury, chemical irritants, and pathogens. Tight junctions between keratinocytes help seal the epithelium and reduce water loss, while desmosomes maintain intercellular cohesion during stress or friction. The surface of the skin is slightly acidic due to the combined effects of sebum and sweat. This acid mantle inhibits the growth of many pathogenic bacteria and fungi. Sebum, secreted by sebaceous glands, contains fatty acids and antimicrobial lipids. Sweat, especially from eccrine glands, contains antimicrobial peptides such as lysozyme and dermacidin. Together, these create an inhospitable environment for pathogens. The skin is home to a variety of immune cells, which serve as sentinels and responders to microbial invasion or injury. We discuss these later in the immune defense section. Sunlight, particularly its UVB component, can directly damage DNA in skin cells. When the damage is severe and repair mechanisms fail, mutations may accumulate, leading to skin cancers like basal cell carcinoma and melanoma. To counter this, melanin, especially eumelanin, absorbs and scatters ultraviolet B rays. This protection is particularly important because UVB can cause thymine dimers. These are abnormal bonds between adjacent thymine bases in DNA, and they are one of the earliest mutagenic changes that can lead to skin cancers. The skin hosts a community of commensal microorganisms known as the skin microbiota. These non-pathogenic microbes compete with harmful bacteria for space and nutrients, making colonization by pathogens more difficult. In addition, they contribute to immune modulation, helping the skin learn to distinguish harmless antigens from dangerous ones, a concept known as immune tolerance. Thermoregulation is coordinated by the sympathetic nervous system, which constantly monitors and adjusts core body temperature. In heat, eccrine sweat glands are activated to release a watery secretion that cools the body through evaporation. Cutaneous blood vessels undergo vasodilation, increasing blood flow to the skin surface and enhancing heat dissipation. This also explains the flush appearance during overheating. In cold, vasoconstriction reduces blood flow to the skin, conserving heat by minimizing loss to the environment. This results in pallor, especially noticeable in the extremities. Why is it dangerous for blood vessels in the dermis to stay constricted for too long? Prolonged vasoconstriction, particularly in freezing conditions, can cause ice crystal formation in tissues, potentially leading to frostbite. The skin is richly innervated with specialized sensory receptors, collectively known as afferent nerve fibers. Sensory receptors are classified based on their adaptation speed. Rapidly adapting receptors respond strongly to changes in stimuli, but quickly reduce their response if the stimulus persists. Slowly adapting receptors continue to respond as long as the stimulus continues. Can you think of a real-life example that illustrates the difference between rapidly adapting and slowly adapting sensory fibers? The moment you put on a shirt, you feel it briefly, but then the sensation fades even though the shirt stays on. That's rapid adaptation. When you're holding a pencil, you constantly sense its shape and pressure. That's a slow adaptation. So let's look at key sensory structures. Hair follicle receptors. These are rapidly adapting nerve plexuses that surround hair follicles and detect light touch and hair movement. Free nerve endings are found in the epidermis and apodermis. These are the most abundant and simplest receptors. They detect pain, temperature, and crude touch. Unlike other receptors, they are not encapsulated or associated with any accessory structures. Meissner's corpuscles are located in the dermal papillae of glabrous skin, especially fingertips and lips. These encapsulated receptors are finely tuned to light touch and low-frequency vibrations, helping detect texture and motion. Located deep in the reticulodermis and hypodermis, there are Pacinian corpuscles. These resemble onions under the microscope due to their concentric lamellae. They are rapidly adapting and specialized for sensing deep pressure and high-frequency vibration. In the deep dermis, Ruffini corpuscles are slowly adapting receptors that detect skin stretch, sustained pressure, and proprioception. They also contribute to detecting heat. And finally, Merkel discs, situated in the basal epidermis, especially in fingertips. These are slow adapting and contribute to fine touch, pressure, and shape discrimination. The skin is equipped with both innate and adaptive immune mechanisms. Let's break it down into three core components. Cytokine signaling occurs in response to injury, infection, or allergens. Keratinocytes and resident immune cells release cytokines such as interleukin-1 and tumor necrosis factor alpha. These molecules initiate inflammation, recruit immune cells, and stimulate tissue repair. Antigen presentation is a process in the immune system where fragments of antigens are displayed on the surface of antigen-presenting cells and macrophages to activate T cells. Barrier immunity, as a part of innate immunity, refers to the preformed defenses of the skin, including keratinized layers that prevent pathogen entry, acidic pH, antimicrobial peptides, and the commensal microbiome, which competes with pathogens. Which immune cells contribute to the skin's defense mechanisms? Langerhans cells, found in the stratum spinosum, are a subtype of dendritic cells and act as antigen-presenting cells. Dermal dendritic cells also act as antigen-presenting cells, particularly in the deeper skin layers. They capture foreign antigens and migrate to regional lymph nodes to activate T cells. Interestingly, keratinocytes can be considered as immune cells because of their cytokine secretion. Macrophages located in the dermis have several roles, such as phagocytosis and antigen presentation. Mast cells play a key role in allergic reactions by releasing histamine. T lymphocytes in the skin are mostly resident memory T cells, which allow the skin to mount rapid and antigen-specific responses upon exposure to pathogens. The skin plays a pivotal role in the production of vitamin D3. When exposed to UVB radiation, 7-dehydrocholesterol, a cholesterol derivative, is converted into cholecalciferol or inactive vitamin D3. The cholecalciferol is transported via the bloodstream to the liver, where it's converted to 25-hydroxyvitamin D3 or calcidiol. Finally, in the kidney, it undergoes hydroxylation to become 1,25-dihydroxyvitamin D3. The skin participates in the excretion of water, electrolytes, and metabolic waste products. And that wraps up our exploration of the skin's physiology. To help you consolidate everything we covered, I've included a detailed mind map right here. If you found this helpful, like the video and consider subscribing to BenMed Academy for more medical videos.